Okay, dear colleagues, let us continue. And it's my great pleasure to introduce Thomas Bosch from the Kiel University in Germany. And actually, Thomas, maybe last time we talked in person, it was 10 years ago. But actually, our meeting in Okinawa Institute of Science and Technology, in the same kind of meeting, same kind of retreat meeting, kind of defined and pushed me to organize this Sochi-based part. So it's another example. So Thomas really motivated us all. So it was kind of 30 young scientists, and his talk was one of the most motivating. That we should study some kind of non-model stuff, some crazy questions, some unusual animals. And I'm so happy that he agreed to talk to us today. So it's a morning in Germany, it's a Saturday. So Thomas, thank you very much, and please start.
Thank you very much, Oleg, and also Igor for the invitation to this unusual meeting. I think it's a wonderful idea for the junior generation to open some windows and inviting senior people to show what kind of windows there are to look into. Since this is a meeting for the junior generation organized by Oleg and Igor, I decided to give you a kind of a more historical perspective of how can you make your life with the untrodden path, an unusual model system. And just this morning I went back and I show you, I'm with this model since nearly 40 years, and I was introduced to it by Charles David. Charlie was the last student of Max Delbrück, and when Charlie came to Munich, he brought Hydra. And that was the decision for my life. And I never changed it. And I got all my jobs and positions and responsibilities because I used this simple model system. And I'm very happy, and I was pleased to see when Alexander talked just right now to how this model still has, I think, a great future. So and I don't have to introduce the model anymore. It's a very simple metazone and has only 2 cell layers, and in principle looks pretty much like a gut. And that's what we are studying for the last 40 years or something. And the major question, which is a fundamental biological question, is of course, how did complex animals evolve? How do they function? How do they originate? And so the question is how to maintain a complex structure over time, over a long period of time. And that can be an individual lifetime, or that can be evolutionary time. And I have a deep affiliation with Russia, as you may know. I'm holding an honorary degree of St. Petersburg State University. I love to travel through Russia. Some years ago, I was at the very north of Russia in the Zoolovsky Monastery at the White Sea. It was a remarkable week where we just drove around and I learned that this wonderful and historical place, to maintain that structure, it has to repair and to repair and to repair. And wherever you go, you find places which are under construction. And I'm very sure this would be it will be a wonderful place when I return back, maybe in some years. Repair and preserve as good as you can. That's what stem cells in our body do. We are born and then we have to function. And most of our organs, and Igor Adamaiko introduced you some of them, including the nervous system, must get repaired over our lifetime. Otherwise, we will not function. And the same is true for the gut, and the same is true for the skin, and the same is true for many organs. That means it needs stem cells. That was the question of my life. For the junior generation, the first and important thing, and Igor was asked the same question earlier this morning, find your right question. The question which intrinsically interests you. The question which keeps you away from sleeping at night, what I call it. Because only then, if you found the question, you can be good. And good means you have to work hard. You have to be persistent. Success may come or may not come, but it's your life and it's your question. And what I will go now in the next 20 minutes is I go a little bit through my life and show you what we have done with many different people at many different places. Studying from Germany to the US, to the UK. I have, as I said, a very close collaboration and affiliation with St. Petersburg. Alex is one of the people from St. Petersburg State University. I had many before. And today I'm working very multidisciplinary with many different people, just again, to very basic fundamental questions using this simple model organism. But of course, developing technologies, omics technologies, developing the transgenics, it took me 10 years to molecularize the model. We developed transgenics in 2006, I think, finally. I spent all the Humboldt Foundation postdoc fellowship in the US by failing to get transgenics. And only with a fantastic technician in Kiel, we finally managed to get that. So I would like to walk through a little bit to that, and it may give you an idea how to shape your own career, and what's important and what's not so important, and how at the very end you always see, when you invite senior people, you see glossy stories and all, textbooks. That's great. It didn't start like that. My PhD, by cloning stem cells, for 2 years, nothing worked. But then we got it. My postdoc Humboldt fellowship, for 1.5 years, nothing worked. Actually, the goal, transgenic hydra, we didn't manage. But still, at the end, it worked. So be persistent. Stick to your question. And on the way, you will find beautiful answers at the side track or at the main track. And it's all depending on people. And I will cite the papers, and I do not mention all the people individually which joined my lab in the last 40 years. But Igor said also a very nice word earlier this morning. Hire people which are smarter and better than yourself. That's the success. I am very happy to be surrounded by fantastic junior co-workers. And you need money. University doesn't pay you, which allowing you the money. That's not enough money. You need external money, and you need it throughout your life. So throughout my life, I'm very much happy to be supported by the German Science Foundation, by some US grants. I started with a Humboldt postdoc fellowship and different sources. You have to continuously write grants. For that, you need to continuously write papers. When you have papers, you may write grants again. It's the scientific, the academic circle. That's our new building where we do the sequencing technology, and that's where I'm sitting in the 4th floor of the Zoological Institute. So I would like to start why we got to this simple model system and what did we learn at the very beginning. And as I said, stem cells are the crucial component of an organism to maintain its structure over time. Stem cells are also important for ontogenetic development, for embryonic development of the organism, of course. Hydra has a number of different stem cells. Some of them are multipotent, others are unipotent. And over many years, we learned the molecular language. We developed, as I said, a transgenic technology, which is now worldwide used to not only to visualize the stem cells. Here you see GFP expressing stem cells crawling around and getting distributed in the newly formed bud of an animal. But we also can now manipulate the cells. Alex Klimovich also developed beautiful technologies. We can ablate cells now genetically. So we can apply the whole molecular tools to understand the stem cell biology in these organisms. And we learned the molecular signature. We learned that there are transcription factors which are very conserved in the animal kingdom among many different organisms, and this is a basic core key to transcription factors. And among them is an interesting one, and that is FOXO. FOXO is one of the major components of longevity in humans. And Oleg sits in Okinawa, and in Okinawa they have a very old, they have centenarians and people which get very old, and many of them may have a certain FOXO allele, the FOXO3A variant. Hydra continuously produces and doesn't age, and for that reason it was so exciting to find FOXO as one of the major transcription factors in the animal. So that's what we learned some years ago and got quite convinced, well, these animals have a very good conserved stem cell machinery, a molecular machinery, which keeps these animals going. Very recently, we expanded that by showing that the segmentation, the spatial arrangement of stem cells along the body column, and stem cells are exclusively localized in the body region and are not in the head and not in the foot, is due to some epigenetic regulation, histone acetylation. We can genetically disturb that histone acetylation, and then we can transform an animal in a purely gastric something, has no head and has no foot anymore, because we destroyed the boundary. The boundary is maintained by epigenetic modulation. The big question now is, what regulates the epigenetic regulators? I come back to that later. So that is the stem cell history of the model system Hydra. I got very early interested in the question of the evolution of the immunity and the question, how can these simple animals survive in the nature without having a skeleton, without having a cuticle, without having anything? It's just a glupsy thing of 100,000 epithelial cells. So it must have a fantastic, efficient immune system. And maybe our human epithelial system is based on what our ancestors have developed. So let's find out what is this defense strategy in these ancient animals, and maybe then we can use this knowledge also for medicine. And what we found is that, so there's a classical toll-like receptor pathway which has TLRs, and they get stimulated by bacterial components, and that gets transduced, and after that, that activates then antimicrobial peptides, and these antimicrobial peptides control and deal with the microbes. All of that was possible with genetics and with knockdowns and with omics technologies. And so we were very pleased to find this ancestral pathway here and to learn how our ancestors did the job. No finding in science is just very straightforward. Usually you get unexpected results. And an unexpected result that I will show you comes a few minutes later, and it brought me to rethink the role of the immune system. And I come back to that in a minute, because we discovered that the Hydra is colonized as any organism on this earth by a stable microbiome. And that was in my personal life, and I can tell you the day when I realized that this is significant, it was 2011 in a conference in the US. This changed my thinking. It changed the thinking of the role of the immune system. It's changed the thinking how regulation is done in terms of stem cells and environment. It changed how we think about the nervous system, and Alex told you about that already. But this was only possible in the 2010s because novel technology allowed us to deep sequence tissue and to find that all epithelia, which we brought to our next door lab and let them get sequenced there, was contaminated by microbial sequences. And it turned out that this contamination is not contamination, but real colonization. And the whole animal is colonized by a very stable, species-specific microbiome, as any other organism on this world. And that brought a completely new side to our research. And what we now know is that not only Hydra, but the organism is multi-organism. You are many. You have at least the same number of bacteria in your body as your own cells. You have more than 100 times more bacterial genes in your body than your own genes. And we consider organisms as meta-organisms or holobionts. And what you see here is in this animated movie made by a collaborating public outreach company, science communication lab, is that this microbiome changes with life stage. And a juvenile has a different microbiome than an adult. And this is functional. And colleagues at the Max Planck in Cologne have shown that you bring juvenile microbes in an adult body, the adult body get juvenized. So the microbes have a tremendous function. The microbes and the microbiome, the sum of all the microbes in our body, is the term for the invisible component of our world. And just a question which was brought to Alex some minutes ago, I can answer very briefly. Why is that so important? Can Hydra not do without microbes? No. In nature, there are no germ-free animals. Microbes were first. And the life was about 4 billion of years full of microbes. Archaea, bacteria, viruses, fungi. And only about 560 million years ago, more complex multicellular plants, animals, fungi developed. They developed on a biofilm of microbes. The microbes have learned to interact, to communicate antagonistically, synergistically with each other. So life and mechanisms of life were deeply and first invented by the microbial world. And then the first eukaryotic cell came on this earth. And it happened that it only was by phagocytosis or engulfment of a bacterium that became a eukaryotic cell with a mitochondria, or maybe even with a chloroplast. All of that happened only because the microbes were first. So it doesn't really astonish you that many, not all, but many life processes can only be understood when we think in microbial terms. And I would like to tell you, health and disease is multi-organismic. And only if we consider health from this kind of holistic point of view, or disease, we really can understand fundamental and complex diseases. We have to take the microbes into account. That was the major change. So we developed the holobiont imperative, which we think needs a holistic understanding of complex life processes. I'm leading a research center since many years, financed by the German Science Foundation, called Origin and Function of Meta-Organisms. I'm a member of a Canadian Institute of Advanced Sciences, Human and the Microbiome, and we try to understand the functional interaction between the microbes and the host tissue. And I take Hydra as a model to develop the fundamental concepts. We consider organisms as meta-organisms. This was how you learned when you are a zoologist or a botanist or a medical doctor, how to look to your subject. This is the red spot. But modern technology in the last 20 years has shown us that this is not enough to understand the function or the dysfunction of this host. You have to see it in the context of the other organism this organism is stably associated with. And any disruption of this association, symbiosis, causes problems, dysbiosis, disease, chronic inflammatory diseases, increasing these days in humans tremendously. Adipositas, Crohn's disease, allergies, neurodegenerative diseases, increasing because there is a disruption of the microbiome. We disturb the microbes by our lifestyle, by antibiotics, by our diet, by many other things. In Hydra, the microbes are living outside in a glycocalyx, which is similar to your mucosa in the intestine. It's a complex microbiome, very stable, many different sorts which are living there, and they deeply influence the functionality of the organism. And that brings us back, we evolutionary developmental biologists, to Van Weyl, who many years ago has already said it's not enough to understand evolution and development. You have to understand the ecology of evolutionary developmental biology, because evolution is the control of development by ecology, by the environment. And so we strongly advertise, together with Scott Gilbert, that we need an ecological understanding of developmental biology. And yes, there are many instances and cases when you now dig deep into the developmental biology of Hydra, that you can find influence of the microbes on the host. And one example is, if you go to pattern formation, you can disturb the pattern formation by disturbing the wind signaling pathway. And this disturbance is very much dependent if there are microbes present, control, or if there are microbes absent, germ-free. So the microbes influence how the wind signaling transduction cascade is actually communicated to the individual cells. Animal development happens in a microbial world. Symbionts educate the immune system, in Hydra as in humans. If newborns are born sterile because they are by cesarean section or the mother took too much antibiotics, their T-cell population will be very different from normal kids. And statistically, cesarean section born kids have a medical and a disease-prone life prognosis. Symbionts affect neurogenesis, that they affect development, growth, and maturation. Of course, development happens without symbionts, and we have germ-free facility and thousands of mice living without germs artificially in our mouse facility. But they have defects in the immune system, in the development, in the nutrition, in the metabolism. Because in nature there is no germ-free animal, and we have to understand what is the function and the contribution of the microbes. Let's go back. I told you FOXO, an important transcription factor from Hydra to humans. FOXO talks, and that was a major surprise by a wonderful graduate student, Annemarie Böhm, published then in the PNAS in 2012. She found FOXO, but she looked for the target genes of FOXO. And she found that the target genes of FOXO are antimicrobial peptides. And in the meantime, we learned that antimicrobial peptides regulate the microbiome. So the basic stem cell machinery is not working in a sterile bubble. It talks to the environment. And we have evidence that the environment talks to the stem cell machinery. Please consider animals do not develop and function in a germ-free environment. And when we do not take into account the environment, we will never understand at a deep level the fundamental processes which are governing these life processes. Very shortly, and Alex brought you a beautiful introduction, what we have learned in the last few years on the nervous system. That was another major surprise, which went back many years. I had a Japanese student in the lab, and we had germ-free animals, and they had a very distinct antimicrobial activity. And at that time, sometimes you publish papers where you have no interpretation for the data. And of course, this then can be only a minor paper, a minor journal, but we published that. Only now we understand that neurons produce antimicrobial peptides. The red little spots here are the bacteria, the green is the neurons, and the blue is the nuclei. And in these early days, 20 years ago, longer than that, I guess, a student from LA, we found that in the absence of nerve cells, there's a very different antimicrobial activity than to controls. Of course, now we know, Alex found out and others, that neurons produce antimicrobial peptides. Oh, wow, the neurons talk to the environment. Alex showed you this picture. So the neurons that stick to the mucosa, and former postdoc René Augustin has found that the neurons not only stick there, but they secrete antimicrobial peptides. And these antimicrobial peptides control and regulate the microbiome. From that and many other observations in my lab, we come to this interpretation, rethinking the nervous system. That means it's not only for motor, so sensory input and motor output, it is also there from the very beginning, because that is the ancestral, most ancient nervous system which exists on Earth. It's also there to communicate with the environment, the ecology, which is in the most intimate environment for an organism, is the microbiome. Nervous system is built on the interaction of the microbes, and the microbes have learned to talk to our nervous system. So I would really like, and I'm coming to the end, that I would really like to stress. I was president of the German Society of Developmental Biology for some time, and I could not convince my colleagues that environment matters in developmental biology. I speak now to a wonderful young generation of researchers in Sochi, in our audience hall, and I would like to stress, please take the environment serious. You are confronted with complex, fascinating processes which your model system gives you. I can promise you will not understand in depth the answer, how they solve these processes, how they manage them without taking the environment into case. And when I talk to young medical students, I will tell them, you cannot understand health and disease without taking the environment into consideration. That's what we are doing here in Kiel in our medical faculty. We have so-called comprehensive centers of, for example, inflammatory medicine, where we consider chronic inflammatory diseases from a holistic point of view. We must switch from an isolated view of a discipline to a multidisciplinary thinking. I have no idea about modeling, about mathematics, about ecology. I'm a basic zoologist. I know Hydra, but I have invited many other people around me, and we work together to get to a solution of these fundamental problems. And only together we get to that. That's a challenge for the junior generation, because you have to learn to cooperate with people from different disciplines. You have to learn to speak their language. You have to learn to speak a language by yourself which they understand. But at the end, it's very satisfying, because you will see a new world, and that is animal development happens in a microbial world. So from choosing more than 40 years ago the untrodden path to understand how multicellular animals originated and function, we come to many paradigm shifts. What's the function of the immune system? It's not to fight pathogens. It's to manage the beneficial microbiome. What's the function of the nervous system? It's not only in motor output and sensory input, but also communication with the environment. It's a fascinating and exciting new way of exploring development. So, what keeps me from sleeping at night? Still today, I have fun every single day. I enjoy what I'm doing. I find the questions fascinating. I'm extremely proud and happy having people around me who share my enthusiasm. And I think the challenge which we are addressing is big. We're not only trying to understand the functioning of multicellular organisms, but fundamental questions, how life on Earth is organized. And with that, this is where I'm sitting right now, and Alex also. We are in the beautiful city of Kiel at the Baltic Sea. My lab is a little outside here, and as Alex said, we are open for applications for graduate students or postdocs. Write an email and you will get back an answer. And with that, I go back to the trajectory of my life, and I'm very happy to answer questions. Thank you very much for the invitation. Thank you for listening to me, and ask questions if you want.
Thomas, thank you very much. Always inspirational. Okay, any questions or comments? But as always, I have many. Okay, let me start. Thomas, so this is a very deep story of Hydra, but is it the first animal in the line of this kind of microbiota nervous system interactions? Or is it a simple example? Or if we move forward to, I don't know, to other invertebrates, you will see the same picture. So I'm sure you have this knowledge. So how it's going? Because Hydra is a great example, but is it the simplest one, the first in the line, or what is there?
Yeah, thank you so much. Hydra is indeed the simplest organism, which first of all has a kind of a complex nervous system. You know that sponges do not have a nervous system, but they have neurogenic, they have genes of the neural pathway. They have also cells which are responding, but they do not have a nerve net. So Hydra, that's our discovery. We discovered Hydra is the first animal with a nerve net, and this nerve net communicates with the microbes. But that is true for all other invertebrates and vertebrates on Earth. And in our community of host microbiome researchers, we have a term for that, and the term is called the gut-brain axis. And today we know that when we disturb this communication between the microbes and the nerve cells, we develop problems. We as humans develop problems. This is mediated by the gut and actually by one big nerve system, the nervus vagus. And if you do a technology, a medical surgery, a nervectomy, you cut away any gut-brain functions, gut-brain activities. So our central nervous system, as our enteric nervous system, interacts with the microbes. This is very complicated to study. Many people do that. If you go in the Medline or somewhere PubMed and get in gut-brain axis, I guess you get kilograms of papers out.
That's true.
The first ones, and an easy one to study, is in Hydra.
Thank you. Any questions? Okay.
Thank you very much for your talk. Unfortunately, I missed the first part. Maybe you told already about that. But anyway, what happened with Hydra if you keep them growing in completely free microbiome conditions? And that is the first question. In particularly, so they're still alive, for example, or you just see the effect of microbiome, but not so crucial on development? And the second question, I have the second question just immediately.
Okay, just let me answer the first question. Okay. So we have, these studies are possible only because of major technological breakthroughs. And this is not trivial. The one breakthrough I mentioned, this is sequencing technology. We first realized that these microbes are there because of deep sequencing. Now we can visualize with super high resolution microscopy, we can visualize the individual microbes on the tissue. But for functional analysis, what are these microbes doing? You need exactly what you are asking. You have to keep these animals in a germ-free, in a bacteria-free culture condition. And that's what we do. And that's what Drosophila people do. And that's what mouse people do. I mentioned we have a germ-free mouse facility. I have colleagues in the institute which are working with germ-free Drosophila. And germ-free animals are around since the Second World War. And first germ-free animals developed ever was in Czechia, in Praha, germ-free pigs. So the animals live, they reproduce. I could show you a movie how they do, but they are not normal. And that is the silent and the deep impact of the environment. Of course, you can reproduce and sperm will meet egg and there will be early development. But there are some parts in your development, when the cells, and Igor showed this very nice trajectory of cells, the cell has to make a decision. And because microbes are always there in evolutionary time and also from ontogenetic terms, cells at certain decision-making turning points wait for a signal. And there is an immunoprecursor cell, which will become a T cell at some point. At some point in embryogenesis, it waits for a signal. Because you grew up your mouse under germ-free facility, the signal is not there. So the cell goes in this way. It should go in this way if the signal is there. That's why kids who are born cesarean section have a different T cell population repertoire than normal born kids. So yes, the answer to your question is, and we have long-term, I have a wonderful Chinese postdoc, Jinru He, who managed with lots of effort to keep Hydra now germ-free for years. The challenge is feeding them germ-free. We monitor them very regularly. They have not a single bacterium. Very difficult to manage, but they are wonderful source. We do RNA sex with them. We do single-cell transcriptomics with them. We are preparing a major paper on the signature of these germ-free animals. They are not normal. And by comparing them to normal animals, we can find out the hidden and non-visible contribution of the microbiome. And that we can do in Hydra, other people do it in Drosophila, other people do it in mouse. And people, humans, taking too much antibiotic, they do a natural experiment. Thank you.
Thank you so much. So the second question, are there any other data on microbiome effect in other cnidarians, not just beyond the Hydra, for any other species?
Yeah, I'm sorry to say that not so much known, I guess. I mean, I'm collaborating with the coral community. In corals are much more important. Hydra is unimportant. And I always say, you know, I only learned in my life Hydra, and this is very unimportant. Corals are very important because they are going to die because of the climate change. So my colleagues at the coral community are deeply trying to understand the symbiosis, the symbiotic interactions between the eukaryotic symbiont, which are Symbiodinium, which are photobiont algae, and the microbiome, which is also in the corals. And there is good evidence that there is a tight and intimate relationship. The functional depths, what we can do in Hydra, because we can do very normal genetics now. We can, in both sides, by the way. We can label the bacteria genetically. We can genetically manipulate the Hydra. this is not possible really in other Cnidarian systems. So I'm very happy to collaborate with my current people. I try to help them and I give them ideas. But the analytical depth, I guess, is very difficult to achieve in other Cnidarians. There is Nematostella. This is a very nice model system. There is Crucia in a lab in Villefranche. I still think that Hydra is, in terms of analytical resolution, a very good model system.
Thomas, thank you. And to wrap up, I have a last question, which is not scientific, but rather structural. So we are talking now, so this event is hosted by a newly established university, so-called Sirius, located in very nice subtropical zone, something like OIST 12 years ago. And you carefully observed the development. So do you have any advice to how to, so you're observing, you're a good friend of Russia, you work with Russian scientists, you have Russian students. So if you would be invited as an advisor to kind of give advice how to position the new university, specifically in life science, what would you advise, seeing of how Russia develops? Apart from political, rather structural things, how, do you have anything to say how to develop brand new university in life science direction?
Thank you very much, Oleg. I mean, this is how many hours I can talk to you now.
That's the point.
This is an excellent. This is of course a very good question. I'm very happy to answer. I am an advisor of a number of universities, and I haven't heard about the university. You talk about it. I would be very happy to share my views on that. The first message is very clear. If you establish a new university in this existing world, the most important point is the unique selling point. What makes this place unique? Why to found a new place? So you have to find a program and a research program, a teaching program, which makes you unique. And then you have to look on your resources. How much money do you have? And if you have a limited amount of resources, as I assume you have to focus. Where are you very strong? Where can you recruit? In which subject can you recruit the best people you can think of? And then build around that people the university with a unique selling point and with a unique teaching and training program. Would be happy to help you.
Thank you, Thomas. Thank you very much, and let's thank our speaker. Thomas, great pleasure to talk to you. Let's stay in touch. Дорогие коллеги, thank you. Дорогие коллеги, у нас начинается ланч, который сегодня покороче, чем обычно. Мы надеемся начать в час, чтобы успеть с нашей долгой программой, поэтому будьте уверены, потому что у нас суперинтересные доклады при поддержке Российского научного фонда. И невозвращение к часу может непонятным образом повлиять на ваши заявки в РНФ. Помните это.